Silk cultivation is a cyclical and well-regulated process, and it begins in the grainage room. The grainage room is where healthy male and female moths are selected, mated, and allowed to lay eggs. These eggs form the starting point of silk production. After hatching, the young silkworm larvae are transferred to indoor rearing units. Their entire growth depends upon mulberry cultivation, as mulberry leaves are the sole food of silkworm. So the quality of mulberry leaves directly influences the larval health and silk quality. Inside the rearing unit, the larvae passes through 5 larval stages called as instars, and during this period they are fed with fresh mulberry leaves regularly and maintained under controlled temperature, humidity and hygienic conditions. When the larva stops feeding and begins lifting its head, it indicates maturity and readiness for cocoon spinning. At this stage, they are transferred to montages, which are also known as chandrikas, which provide support for cocoon formation. Inside the cocoon, the silkworm secretes a single continuous silk filament. After the cocoon formation, the cocoons undergo stiffling, which is a process that kills the pupa and prevents the moth from emerging and breaking the silk thread, which will reduce the quality of silk. The stifled cocoons are then processed using reeling or spinning machines to extract the silk filament. From this process, 2 types of silks are obtained: the finer ones used for premium fabrics and the coarser one used for thicker, stronger textiles. Finally, a portion of cocoon is allowed to develop into moths and sent back to the grainage room, where the egg production takes place again, completing the silk cultivation cycle. Silk cultivation is not just a biological process, it is a system of precision, care and continuity.